Hello, everyone. My name is Jun Ishihara. I'm a assistant professor at Department of Bioengineering. I am a collaborator of David Baker, Professor David Baker, and it's very grateful. It's very fantastic to have you here, David. And let me explain David's bio. So Professor Baker is the director of the Institute of Protein Design, a Howard Hughes Medical Institute investigator, a professor of biochemistry at the University of Washington. Dr. Baker has published over 550 research papers, been granted over 100 patents, and co-founded 17 companies. And remarkably, 68 of his mentees have gone on to independent faculty positions. So Dr. Baker is a recipient of Breakthrough Prize in Life Sciences and a member of NAS. And his team developed Rosetta Ford. I'd like to highlight that his work led to the Science 2021 breakthrough AI-powered protein prediction. So thank you for accepting the seminar today, and who is yours?
Okay, well, thank you for the introduction. Thank you very much for the invitation. I'm sorry about the complications getting started. Let me go ahead and share share my screen. Okay, so I was just going to say, so just over the, so I'm going to talk about protein design today. And up until about a year ago, all of the work we did was based on physically based models using a program called Rosetta that we and our many colleagues around the world have been developing. And in that model, we describe all the interactions between side chains and atoms using a physical model. And the basic principle is that proteins fold to their lowest energy state. And so when we are designing a sequence to fold to a particular structure, we search for a sequence whose lowest energy state is that structure. So that then when we make the protein in bacteria or yeast or mammalian cells, it will fold up to the designed target structure. And as I said, this was the approach we used up until about a year ago. And now, more recently, we've been using deep learning methods to design new proteins. And I'll talk about that towards the end of my talk. Um, and uh, um, and so what it's a very exciting field right now because it is it's in transition and things are changing very rapidly. It's also an exciting field because I've been working on protein design for a long time. It really hasn't been all that useful. It's been kind of an academic exercise, but recently that's been changing, and I wanted to highlight that at the beginning of my talk. So we have been designing proteins to bind to the receptor binding domain of the coronavirus spike protein. So these are very small proteins from 65 to 75 amino acids. And we basically designed them to be chemically and shape complementary to the viral spike protein. And we have, I've talked about those extensively in previous talks, and we've published actually a fair amount on them as well. So I didn't want to go into the details of how we do that design. But what we've been working on more recently is, of course, there's been a succession of variants of concern that have emerged, and um, the most recent, of course, is the Omicron. And so what we have done is to design. So the small proteins I was talking about are represented by this red one here. This is a design model. So here we have the spike trimer, and here we have a design which has not just one of these designed binding but three of them. And so this is a designed protein which is a trimer. It's like a tripod. It sits on top of the coronavirus spike protein. And what's neat is each of these small proteins mimics or binds to the receptor binding domain. And this particular protein called AHB2 makes very similar interactions with the receptor that ACE2 does. And So it's very hard for the virus to escape, because if it mutates those residues, it can't get into cells. This is a cryo-EM structure of that protein bound to the spike trimer. And what's very encouraging is, against every variant that we have tested, this compound is very effective. It shows 100 picomolar neutralization efficacy in cell culture against Omicron, and similarly against Delta and the other variants which have been emerging. And so we think it's likely a compound that is very difficult for the coronavirus to escape from. So this compound is currently in clinical development, and we're and hopefully it will be useful in the world. So we're using a similar strategy to make antivirals against other likely pathogenic viruses. So we're making binding proteins to influenza, MERS, and other viruses. And this general concept, that the advantage you get from linking domains together and geometrically matching is there's an increase in avidity, because you have not just one binding interaction, but you have 3 binding interactions. And another nice thing about this compound is, I said that we had interactions that were similar to those in ACE2, but we don't have, but the sequence of the protein is actually completely different from ACE2. And so it's very unlikely that if there are antibodies against this compound, that they would cross-react with our own ACE2, which is important. And again, so we think this could be a very broad and general antiviral strategy. These compounds have been effective in animals when administered into the respiratory system as a nasal spray, and that's the way that they're being currently developed for human use, both before infection and after. So there's a first illustration of how protein design can actually start to be relevant to problems in the world today. So the second thing I wanted to talk about is the design of compounds to this is now showing how protein design can be used for compounds that actually really don't look like proteins at all, and are made of far more than just the 20 amino acids. And so here we've been exploring 2 classes of compounds. The first compounds are cyclic peptides, where the N and C terminus are bonded together, that are between 8 and 12 amino acids, that are made out of L amino acids, D amino acids, and other types of amino acids, for example, with N-methylation. And we've been exploring ways to principles for getting these across membranes. And using the same kind of principles that we've been using for protein design, where I told you we can now control the positions of by designing for low energy states, we have very high atomic accuracy, and that's illustrated here. Just shows comparisons of structures and design models for quite a number of different peptides of this class that we've designed. And you can see that even though you think of peptides as being floppy molecules, we can actually, on the computer design sequences that will fold up to these very rigid structures, and do this with very high accuracy. And what's neat about these is that they are indeed membrane permeable. So this shows permeability in an artificial membrane system, where we're measuring the movement of compounds from one side of this membrane to another, and you can see that these compounds, a permeability of 1 is sort of the threshold, and you can see we even have compounds out as many as 12 residues, which are getting across membranes pretty readily. And this is because we can program their structures, and we can incorporate into the design process considerations of permeability. And so, we're now basically using these compounds, incorporating binding sites on them as well. And so the idea is, the hope is to make therapeutics that can target intracellular targets or be orally bioavailable. So now, in what I just described, we were still using the standard sort of NC alpha C backbone, but we can go beyond that to use a very wide range of different types of monomers. So this is work from Patrick Salveson, a postdoc in the group, and he's now generalized the protein design, this sort of design machinery, so that we can consider all these different types of chemical backbones. So normally you'd have N, C alpha, C, and here you see we have all these other atoms in between. So there are a lot of, just even at the chemical level, there are a lot of monomer types and a lot of dipeptide types. And so Patrick developed ways of enumerating the sorts of the closed configurations you could make from these compounds, and this shows a number of these. So they're again macrocycles. They're much smaller. These are just built from 4 different units being snapped together. But since there's so much chemical diversity at the backbone level, let alone the side chain level, it's really an enormous space that looks like a lot of, and some of these compounds look like natural product drugs, but it's been very difficult for humans to sort of systematically enumerate them. And again, in this case, we can specifically design compounds which are predicted to have single unique states. So we're at a similar stage here. We've solved the structures of quite a number of these compounds, and they come out quite close to the designs, and we're now trying to incorporate binding functionality. These compounds, and it's perhaps not surprising given how small they are, also get across membranes. So we're very excited about these, and both as potential sort of new classes of therapeutics, but also sort of to illustrate that sort of protein design methodology can now be applied well beyond the sorts of compounds that it has normally been applied to. So the next thing I wanted to talk about is coming back to proteins. Now, sort of the fundamental problem of designing proteins that can fold to more than one state. And this is work of Florian Pretorius and Phil Leong. So here is, we've been designing for a number of years now proteins that are made out of simple proteins that are made out of repetitions of a single unit that just is repeated over and over. And one of these is exemplified here. And what we're doing now is to design multiple states of proteins which can adopt multiple states. So here's one state like this, and then here's another one where you can see this top part has rotated around this helix and is now on this side. And so we call these hinges. And Florian has designed this one so that there's a peptide binding site that's exposed in this form, but not in this form. And when you add peptide, it drives it into this, this state. And so this system can be evaluated in a couple different ways. First of all, we can, by specifying the conformation, we can control whether or not the peptide binds. And by adding the peptide, we can drive the system from this state to this state. And so first of all, this is a crystal structure of the this designed protein bound to the peptide. And you can see it's binding where it was supposed to. In gray is the design model, in color is the crystal structure. So this is first the conformation controls binding case. We can use disulfide bonds to lock this protein in the closed state, in one state or in the other state. And I won't take you through these size exclusion column profiles, but basically the peptide binds only when the protein is locked into the open state, but does not bind when it's in the closed state. And just this, for those of you used to looking at SCC column traces, this is the peak for the complex, which is shifted to the left in the open state case, but not in the closed state case. Now, to look at the driving of the conformational change, we collaborated with Stefan Stoll's lab and used Deer spectroscopy. So in the unbound state, these are the positions of two spin labels that have been put on the protein. And what happens when the peptide binds is they get pushed apart. You can see that schematically here. This is the bound state. This is the unbound state. And if we look at the experimental data, which is here, we see that in the closed state, these spin labels are closer together. And when we add the peptide, they move further apart. And of course, I showed you the crystal structure with the peptide bound where they're further apart. So, so we can use um, uh, we can now design proteins that undergo conformational changes in the presence of ligand. And what we're working hard on now is to use this sort of principle to drive uh, to basically drive a machine. So have a system in which the added ligand drives a conformational change, and then it gets hydrolyzed, so trying to make hydrolyzable versions of the peptide so that it undergoes a full cycle for walkers and rotors. Now, on the peptide binding side, I just wanted to briefly mention another exciting project being done by Keija Wu in the lab. And probably many of you are familiar with the idea that armadillo repeat proteins bind to peptides, repeating peptides, and Andres Plukland's group has done beautiful work trying to extend the specificity with which these peptides can be bound. And what Kate just set out to do is to design repeating proteins that could bind a repeating peptide sequence. And she was she and Hua Bai, who she worked with, succeeded in designing proteins which bind to a variety of different peptides with a tripeptide repeat. Um, and we're very excited about this because while we had done a lot of work on designing proteins to bind to other proteins, we're starting now to be able to make design proteins, as I've shown you in this case and the preceding case, that can bind to much more flexible targets like peptides. And we think with this approach, we should be able to design proteins ultimately that can carry out that very general peptide recognition. Um, So now, switching gears a bit, we've also been interested in designing expandable sort of nanomaterials, proteins that will self-assemble into larger structures, not just bind to peptides, for example. And so here we've designed, and to do this, and to make it really modular, Tim Huddy designed a set of proteins which have uh, the interesting property that they're completely straight. So this is a repeat protein, like I described before, but it doesn't bend or twist at all. So you can just add on more repeats to grow it or shrink it. Um, and so when you build things out of it, like this square, which doesn't really look like a normal protein, but we can do things like this now because we can make these really idealized proteins. Here's the EM density here, and um, we can grow this square out just by adding additional units. So this becomes like, this protein is now like more of a building material that you might have if you were constructing some thing in the real world where you can make things, the edges longer or shorter, which is kind of hard normally to do that with proteins. But here we just add in additional repeats. And you can see in these averages that I think the negative stain is a little bit hard to see, but that the square is expanding and contracting. So working with Yang Xia, Tim took this into 3 dimensions. So now this is a tetrahedron that is built from these very straight proteins, and um, you can see by expanding simply inserting repeats, we can grow this structure out from there's a small size, a medium size, and a large size, and here's the corresponding EM maps. So this is sort of we can not only design these assemblies, but we can design scalable assemblies. Now, um, I wanted to, I think I did show this slide in my talk last year, but I wanted to show another way in which we can sort of build up on top of already existing assemblies. So we described these assemblies last year. These are designs now of different geometries, but they have the interesting property that what we've done is to incorporate into the assembly as a building block a widely occurring protein, basically an antibody, and this can be any antibody. So here we have a pentamer that is designed, that interacts with antibodies with at just the right angles so that the whole structure folds up into an icosahedron. And here's another design protein. These are all made by Robbie Devine. With the antibody
David?
combining site. Sorry?
Oh, we could not hear you for 10 seconds.
Oh, okay. Let's see. Should I start at the beginning of this slide?
When you started explaining about, it's just 10 seconds.
Okay. Yeah. So I was just saying, so I'll just summarize. Yeah, there may have been internet problems. We have designed these proteins to assemble antibodies into different shapes, and in these particles, the antibody combining sites are sticking out, so they can engage the targets. And the nice thing about these designs is any antibody can be assembled into these particles. And so here are pictures of them. And so what we find is when we take antibodies against cell surface proteins, in this case, death receptor antibodies, and we add these particles, we get very pronounced agonism so this antibody alone has very little effect on cells, but um, uh, when it is assembled into these particles, you get clustering of the corresponding receptors, and in this case, cell death. So the reason I wanted to tell you about it today is we can take these particles, and they're made out of two components. This is the octahedral component, which if you think about a cube, it has a fourfold symmetry axis. And I showed you this, it's this one here, which has this fourfold symmetry. And then the antibody sits on the twofold symmetry axis, which is like the edge of a cube. So we've got one protein in the center of the face of the cube, that's this one, and one protein that's on the edge. But there's a remaining symmetry axis, which is basically like the corner of a cube, which if you think about it, has threefold symmetry. And this was empty in this design. So what Aaron Yang did was to design a protein which has threefold symmetry, which sits on the threefold symmetry axis of this octahedron. And she designed this to be pH dependent. So it comes apart at low pH. And so the resulting structure looks like this. So we have an antibody, which can be an arbitrary antibody here. We have the tetramer here, and then we have this trimer on the symmetry axis, which on the threefold, which comes apart at low pH. So we're very excited about this for packaging and delivery because we can target these particles based on the antibody. We can package things inside, and then these yellow domains will come out at low pH. We can also design assemblies that are made out of other functional proteins. And this is work from Nate Ennist and Shunji Wang. So this is a protein which binds chlorophyll that Nate designed. So it's a dimer, and it binds two chlorophyll molecules in exactly the positions they have in the photosystem special pair. And in fact, there's exciton coupling between those two chlorophylls. So what Shunji did is took this chlorophyll dimer and then designed a trimer that could sit on the corner of the cube and assemble this dimer into a cube. And you can see here in the negative stain images, you can see these cubes. And here are 3D reconstructions showing, again, that we can take a functional protein and assemble it into a three-dimensional structure. And this is we're excited about this now for building artificial light harvesting systems because each of the edges of this cube have two chlorophyll molecules. And another area that we have been very interested in is enzyme design. And Andy Ye and Chris Norn decided to take on the challenge of designing a completely new luciferase. And and this is the first appearance in my talk of deep learning methods. So the first step was what Chris did was to generate very large numbers of proteins with pockets with this overall fold using a rapid deep learning generative method. Um, and then Andy used these and then computationally searched for proteins that where we could design side chains that would create a very shape complementary pocket for the substrate, which is shown in yellow here, which would hold it up against an arginine residue, which would promote the formation of an anionic species, which has been implicated in luminescence. And you can see some of the sort of the bottom line data here. So this is just a tube with with a blank with nothing in it. We had an initial design which had luciferase activity, and then with just two mutations that were kind of increasing packing on the substrate, we have a protein which is very, very active. It turns out to has a kcat over km about 10 to the 6th. It's as active as naturally occurring luciferases, but unlike naturally occurring luciferases, it's extremely specific for its substrate because it has a very tight complementary pocket. And so these are different luciferin substrates. And I'm not showing you here the specificity of naturally occurring luciferases, but they act on many different substrates. And here, this designed one only acts on this one substrate. So it's pretty neat. We can now multiplex them and do things that you can't really do with naturally occurring luciferases. And this is sort of this is the that we're using this principle now to design a wide variety of enzymes. Okay, so now I want to get to um, the, uh, um, the deep learning part. So as I said, everything we've done so far that I've told you about, it's been based on physical models where we're modeling all the interactions between side chains. And so, for example, in the case of that switch where we had two different states, we're explicitly looking for a sequence which has two different energy minima. Now, as you know, there's been a lot of progress in protein structure prediction using machine learning. And we've been inverting this to design proteins. And the basic idea is if you have a network that takes amino acid sequences to three-dimensional structures, you can invert this by starting with a random amino acid sequence, putting it through the network, evaluating how well it folds, and then optimizing the sequence so that it's strongly predicted to fold. And we call this deep network hallucination because it's analogous to taking a network that can predict whether an image can basically classify images, for example, cats as cats. You can use that type of network to generate random images and then optimize them to the network really thinks it looks like a cat. And then at that point, they do look like cats, but they're not any actual cat that the network that it exists in the world. It's just a cat that the computer came up with. And we're doing exactly the same thing here, but we're making up proteins. Um, so if we actually do this, and this is a contact map, so showing you residue by residue, we take a random sequence and predict its structure. There are very few predicted contacts, but as we optimize the sequence, there are more and more contacts predicted until at the end, there are many, many contacts, and these correspond to the structures shown here. So we have made many of these computer hallucinations in the lab, and many of them fold up into stable structures. And we've been able to solve those structures by NMR and X-ray crystallography. And they come out very close to the hallucinated structures. So basically, just by starting off with completely random sequences and then having using the model to find so basically this hallucination approach gives well-folded proteins. So this actually was published. I think this is the only thing I'm talking about today, which this came out last month. So now what we can do is, in that case, we weren't putting any constraints on the design sequence. But what we can do is add on additional terms which describe the formation of a particular functional site. So say we want to create a protein that has a particular motif that, say, catalyzes a chemical reaction or binds a protein target. um, we can um, we can basically reward not only as we're optimizing the sequence, the folding to some structure, but also the presence in that structure of the desired motif. And we call this constrained hallucination. And so we can do different things. We can reward the formation of an active site or protein-protein interactions. Um, um, and I'll just show you a few examples. So uh, let's see, let me go down here. So we can take, for example, epitopes from a virus like respiratory syncytial virus, and we can use this approach to come up with scaffolds that are predicted, sequences that are strongly predicted to fold into structures that have these epitopes. And you can see some examples here. And this is similarly with a different epitope where, um, um, so we can basically use this process to come up with sequences that display a particular specified in advance epitope for the immune system. And here's now the same thing for example as a receptor trap. I told you at the very beginning about our use of Rosetta to design proteins that display that mimic ACE2. And here's now the use of the deep learning method. We take this part of the ACE2 protein shown here, and then we basically just hallucinate proteins around that helix. And now in the case of enzyme design, this is, for example, carbonic anhydrase, where we have 3 histidines that coordinate a zinc atom. So we can specify this motif, and then as we're optimizing sequences, we can we reward those that form the site, and so the sequences we get in the end are predicted to. You can see this. This is basically the prediction using AlphaFold, taking that sequence. So we design it using Rosetta Fold, which I'll talk about in a minute, and then we confirm independently with AlphaFold. And we can use a similar approach to design proteins that bind to targets. Now, there's a second approach which we've been developing to design proteins around functional sites, and this is analogous to if you're typing into a word processor, it will try and complete your sentence, and often it can do it pretty well. And so it's basically using a language model, a model of the sequence of English if you're writing in English, and so it can anticipate from a few words what the rest of the words should be. And we can do the same thing with Rosetta Fold, which is also a general model of we've trained to now be a general model of protein sequence and structure. So what we can do is take a partial sequence and partial structure, put it through Rosetta Fold, and have it automatically complete it in the same way that the word processor would complete your sentence. And there are quite a number of different problems we can now do with the same network. We can give it sequence, and it will predict structure. That was how we originally trained Rosetta Fold. We can give it structure and have it predict sequence. That's the sequence design problem. And we can give it just functional sites and have it fill in the rest. So as an illustration of this, so we can take, so if we train Rosetta Fold to predict sequence from structure, again, that's not what we trained it for originally, we get a model that predicts sequence much more accurately than the original Rosetta Fold did. And it turns out not to hurt structure prediction at all. So now we can do neat things like take a protein and remove different parts of it, as shown here, and then get these these sort of truncated structures, and then just give them to the model, and it turns out to be able to fill in the missing region remarkably well. And so we can now do something very analogous to what we could do with hallucination. We can take functional motifs, for example, this is a calcium binding EF hand, throw at everything else, and then the program will generate new proteins that, and you can see this one is, you can see this thing, what's generated here is very different from what we started off. We take a native protein motif, we throw away everything but the motif and then grow out grow out a protein that has that. And this is a similar thing for binding. We keep the binding site. We get rid of everything else. This is from the IL-10 cytokine, and then we can grow back a miniature protein that has that. So we're excited about these new methods. This method in particular is very, very fast because it's just one single pass through the Rosetta fold network is completing the structure. And so we're currently experimentally testing these designs. It's a very exciting time in the lab because everyone's experimenting with different ways of using these methods and combining them with Rosetta, the physical model. And so there's a lot of things cooking right now. So the final thing I wanted to mention, and we also published this recently, but I'm very excited about it, is we took Rosetta Fold, which we had developed for protein monomer prediction. And when we did that, we found that the way we had trained it, it was also quite good at protein complex prediction. And we developed a version of it that was fast enough that you could basically go through pairs of proteins in an organism and essentially go through all pairs and assess whether they were likely to interact. And then as to actually build so we had that first filter, and then we used AlphaFold to put to build models of those complexes we identified that were predicted to interact. And we found that the combination of Rosetta Fold and AlphaFold gave us much more accurate predictions. And when we did this, we did this for the yeast proteome. So we took all pairs of proteins and asked whether and predicted whether or not they interacted. And we identified many new complexes that were spread out in many different functional areas. And some of these are shown here. It was actually a lot of fun because we collaborated with many people around the world that were experts in these different processes, and we sent them the models, and they, looked at them and were able to make some get some biological insights from them. I think it's kind of an important lesson, like these big data or deep learning methods can generate a lot of results, but human experts are still really important for figuring out what they mean. I just want to zoom in on one example. So we built not only assemblies with 2 components, but assemblies with up to 5 components. And here's a 5-component assembly. These are proteins that were all previously implicated in GPI anchor synthesis, but there was no structural information, so it was really not clear on how this complex worked. So we found Tamara Hendrickson, who'd spent many years studying this complex at Wayne State University, and we sent her these models. And she pointed out many interesting features. For example, she already knew that the catalytic site was contained within this subunit, and she pointed out that there was a channel right next to it where the peptide that gets recognized as complex could sit. And indeed, residues that were involved in interacting with that channel were surrounding this pocket. And so this kind of structure obviously now provides a whole bunch of basis for hypotheses for further understanding how it works. So I just want to. So I've had fabulous colleagues who have done this work, and so the Omicron binding work I put in at the beginning. That was work by Andrew Hunt at Northwestern. It's been a big collaboration between many groups. In my group, Longjing Cao designed the mini binders, and Keija Wu designed the trimer, and David Wiesler at the UW solved the structure. And there have been many groups involved in all the animal experiments, and it's currently going through the clinical development process. So for the macrocycles and mini cycles, Gaurav, Patrick, Jacob, and Adam did that work. The peptide-induced conformational change, mentioned Florian and Phil, and then Keija did the work on the repeat protein binding the repeat peptide. The extensible nanomaterial work was done by Tim and Yang, and Robbie and Aaron and Andrew did the work on those 3-component antibody cages. The de novo designed luciferase work is done by Andy and Chris. The protein design using deep network hallucination was done by Yvonne, Sam, and Temuka. Guy Montagnini's group solved the NMR structures. Ming Kyung Bak developed Rosetta fold, and it's continued to develop today. It's actually it's gotten quite a bit more accurate since when we published it. And on my next seminar, I'll tell you all about that. Ju and Sydney and Doug did the did the constrained hallucination, and David and Joe did the in-painting work. And the interaction prediction work was done by Chen, Ian, Aditya, and Ming-Kyung, and then with a host of collaborators all around the world. So thank you very much for your attention, and I'd be happy to take any questions.
Oh, great. So if you have questions, or feel free to raise your hands or leave the comments in the chat. Evgeny, can you speak up?
Hi, can you hear me?
Yeah.
Yes.
Yeah, hi. So I have a question about this part where you're using Rosetta Fold for sampling the sequences. So my understanding of how Rosetta Fold works is that it uses multiple sequence alignments to actually predict the structures, and that and to actually build the multiple sequence alignment from a database, it takes quite a bit of time. So I wonder, how do you work around this issue? Because
Yeah,
presumably to
right. So to build, yeah, there was an important point that I should have made. While for naturally occurring proteins, multiple sequence alignments are required to get accurate structures. Turns out for designed proteins, because basically all the proteins I talked about in my talk, we're building in everything we understand about protein physics. Turns out designed proteins can be predicted very accurately just from single sequences. So in the design calculations with Rosetta Fold that I told you about, we're doing purely single sequence modeling. So because we don't really have multiple sequence information, because the design proteins have no evolutionary relatives. So yeah, so the important point is that the structure prediction accuracy on design proteins is high enough that you can do single sequence to structure modeling.
Great. Raphael, can you speak up?
My question to you is regarding the proteome-wide protein-protein interaction predictions. For example, if you take yeast as a model organism, of course we have many techniques and many years of validation of protein-protein interactions, both obligatory ones and transient ones. And my question to you is with respect to to prediction in yeast, for example, what is the precision and recall compared to those other techniques?
Yes, so we compared to yeast-2 hybrid, large-scale yeast-2 hybrid, and mass spec pull-down experiments. And we had a gold standard. The reason we started with yeast, of course, just for the reason that you gave, that there has been a lot of studies. So we could compare the precision and recall on a gold standard set, on a small set of very well validated proteins. The precision recall were considerably higher than the high throughput methods, which was pretty interesting because it was a purely computational method. Now, the complexes that we identified primarily are obligatory ones, probably. They're not the really weak transient ones. And just to be clear, in comparison to my answer to the previous question, there has to be coevolution. We are picking up in this case coevolution between the different chains, and so if for things that are really weak, that are unlikely to where there really to be much sequence coevolution, it's the methods work much less well. But yeah, remarkably, the accuracy of this purely in silico method is higher than the really high throughput experimental methods.
Thank you.
The next question is from Levan. or should
Can you hear me?
Yeah, sure.
Yeah, I was wondering, is it possible to predict the oligomeric state of homo-oligomers using the Rosetta fold and associated tools? Can you just...
You mean to predict homoligomeric structure?
Yeah, or for example, I have a coiled coil, like for example, those Golgi tethers, which are very long coiled coils.
Yeah.
Can I use Rosetta to find out whether it's a dimer or a tetramer?
Right. Yeah, that's a good question. What I can say is that if you know the oligomerization state, then you can often predict the complex structure accurately. As far as and we haven't really done a detailed study of how well you can predict what the oligomeric state is, and that can be a harder problem because, as you know, there's some proteins that can adopt multiple oligomeric states. Yeah.
And a second question is, with these newly predicted sequences, at least the hallucinated sequences, all of them are expected to fold properly?
Well, when we test them, not all experimentally, so that I didn't really describe the process, but after we have the sequences, we make synthetic genes that encode them, and then we make them in bacteria. What we find is that only and it's true for basically everything I talked about, that I'm showing you the designs that work, but they're always designs which don't express well, the bacteria don't like for some reason. So it's always some percentage. For the hallucinated, for the study I showed you, it was about 30% of the proteins we were able to get soluble monomeric protein from.
Okay, great, thank you. Jiafei, can you speak up?
A great talk. So would it be possible to access somehow the protein dynamics using these deep learning models?
Yeah, that's a really interesting question. I would say that's one of the cutting edge problems. The issue, of course, is that deep learning methods, are trained on static structures right now. They're staying trained on crystal structures, and so deep learning methods are only really as good as the data they're trained on. So, so we're working on ways now for basically having the networks generate a diversity of structures, but actually predicting dynamics, I think ultimately will really be improved by training on actually dynamical data. And there the problem is there just isn't as much dynamical data as there is crystal structure data. So that's one place where, like a physical model, like a molecular dynamics simulation, where it's explicitly modeling motion, for now has advantages. So I think with the deep learning methods, I think we can now generate multiple different plausible states, but actually, predicting what the dynamics is, we're not seeing energy barriers, anything like that.
Great, thank you.
Next question is from Dorian. Yeah.
Can you hear me?
Yes.
Yeah.
This might be noisy in the background from my kids, but I was just wondering, can you just recap a little bit how protein hallucination works?
So basically, yeah, think about the case of the cat example is a good one. So you have a neural network that can take an image and classify it as a cat or a dog, say. And now you can take a random image, and it won't predict it to be classified as a cat or a dog because it just looks like a random image. But now you can start changing that image and keep checking with the network whether it now thinks it's a cat. And at some point, if you sample enough, that it will generate an image that actually the network predicts to be a cat. So it's the same thing with the protein hallucination. We start with a random amino acid sequence. The network doesn't predict it to be a protein at all, to fold up to any structure. But then after we sample through different sequence, sample the sequence space, then it starts to look more and more like a protein. And if we continue that process for long enough, we have a sequence that the network is quite convinced folds up to some structure.
So is it, I'm in the field of cryo-EM, and in that field, we're always trying avoid this whole Einstein from noise principle where the noise builds up to be like the
Exactly.
model, but this is actually what you're trying to do.
That's exactly that, right. That's an even better illustration. That's exactly what we're doing. We're trying to conceive something out of noise. Now, you can understand that there could be problems doing that too, because you could just be amplifying noise. So, but that is basically the concept of hallucination. You're just starting with noise and you're optimizing to get you know, a folded structure. So what we always like to do is to use an independent network then to check, because we might be overfitting against the first one. So if we're hallucinating with Rosetta fold, then we predict with AlphaFold, or if we're hallucinating against AlphaFold, we predict with Rosetta fold.
Great, that's a lot clearer. Thanks so much.
Next question is from Andre.
Oh, hi, can you hear me?
Yeah.
I typed in the chat. I was just wondering whether among any hallucinated proteins there were ion channels or other transmembrane proteins which require lipid. You know, how was lipid energy contribution basically was taking in the optimized function in the deep neural network? That's the question.
Yeah, no, it's a very good question. So we have, using Rosetta, been designing ion channels and now larger pores, and there we have to explicitly consider the interactions with the membrane. But with these deep learning methods, there's no real physics, so it's seeing structures of quite a few membrane proteins, and so it will actually generate you know, channel-like structures, and in those cases, it will actually put hydrophobic residues on the outer surface because the proteins it's seen that have that structure or similar structures have hydrophobic exteriors. So it's you know, it can anticipate things that it's just, you know, deep learning is just doing pattern recognition. So it gets things like that right, but not perhaps for the right reason.
It's like the Matrix.
Yeah, I guess a little bit like that.
Thank you.
All right, Alisa, are you back?
Can you hear me?
Yes.
Thanks for an excellent talk. I wanted to ask about your thoughts on two different protein design approaches that you highlighted in some of your papers, with the first being restrained design around a functional site or motif, and the second being entirely de novo design or from scratch. So if I remember correctly from the SARS-CoV-2 mini protein design, the second approach, the de novo design, appeared to work better. But in this talk, you outlined various designs from a kind of taking that restrained or more rational approach. So I guess I wanted to ask if you have any thoughts on kind of the advantages and limitations, and going forward, if you expect one approach to be more dominant, or if there's definitely applications for both approaches in the design space.
Yeah, that's a very good question, very perceptive. Yeah, so let's focus on the coronavirus. And indeed, if you heard me talk 6 months ago, I would have really highlighted the completely de novo designed protein because the affinity just so basically, if you're given a blank slate and you can bind anywhere on a target, the de novo design approach has the advantage that you can choose the optimal site and get very high affinity. So we got, you know, 15 picomolar binding for just a 55 residue protein. Whereas when we took interactions from ACE2, we were much more limited, and the binding was 100-fold weaker, around 1 nanomolar. However, you flash forward, you know, move forward to today, and we're surrounded by Omicron, and it turns out that as an antiviral, giving up that affinity was worth it for staying right at the ACE2 binding site, where escape was much more difficult, and that then by trimerizing we could overcome that loss in affinity. So I think your answer is a very subtle. Your question is a very subtle question. I think it really depends on the application. I think what we've learned in the case of pandemic viruses that are spreading around the world, that there will be escape, and so a more conservative approach where you try and build interactions that restrict yourself to interactions which are present in the actual receptor can have advantages over a purely de novo approach. So I think in the different areas, the answer is going to be different. I think in general, the de novo approach is going to be more powerful, but there may be reasons why sticking with sort of something that's more based on something we've seen before could be beneficial, as in the in the receptor trap case. So that's a very good question.
Thank you very much. A great answer.
Howen.
Oh, hello, can you hear me?
Yeah.
Yes.
Yeah, thank you, Professor David, for a wonderful talk. Oh, my question is, for the de novo design, when you input the random sequence and the system, the network will show a predicted structure with the lowest energy state, right? Yeah, well, and I still don't understand how you get to the final design. I mean, how do you optimize?
Yeah, well, let's see. So let me just describe the two different approaches. So the traditional Rosetta approach, which is the physical model, you would start with the random sequence and you would optimize that sequence using a Monte Carlo simulation where you would change the sequence, you would measure, evaluate the energy, and accept that sequence change if the energy came down. And so that would be classic molecular system, molecular simulation, where you're basically searching for a sequence whose lowest, which has very low in energy. Now, the deep learning model doesn't know anything about energy. So all it's doing is pattern recognition. So in that case, you start with a random sequence and you predict it. Say you're trying to make an ACE2 mimic. So you predict the structure, and does that predict, and you assess, does that predicted structure, does it have an a binding site like ACE2, and does it predicted to fold up into a well-folded structure? And at the beginning, it won't. So then you start making sequence changes, and you can do this with sort of using gradients traced back through the network or by random substitution. And you just keep assessing that as you go through, and then so you're directly optimizing a sequence that the network will then predict will fold up into something like an ACE2 mimic. So you just keep repeatedly changing the sequence and then asking the network to predict the structure. So the difference between the two approaches is in the classical Rosetta design approach, we're basically evaluating the energy of the sequence, whereas in the deep learning model, we're not evaluating energy, we're just evaluating whether the predicted structure resembles what we would like that predicted structure to be.
Okay, thank you. I understand.
So, Rafael.
Thank you for giving me the chance of a second question. David, I have to admit that I don't know out of the top of my head the accuracy of Rosetta Fold compared to AlphaFold. I do know, however, that AlphaFold 2 is not that very good if we want to predict entire proteins. I like to define as a concept similar to the H-index. And if we think of how many proteins, human proteins, AlphaFold predicted with 90% of its residues above 90 PIDDT, which is a good measure of quality, there are only 800 proteins that are that good of a model, and I don't even know how many of those out of 20,000 could have been predicted otherwise by homology modeling. So, there is a lot of hype about AlphaFold and method of the year and all that, but we should curb our enthusiasm a little bit. Now, you're using Rosetta Fold for full protein predictions and interactions with other proteins. Where does all this uncertainty that still exists? And I'm not against the use of machine learning. I think it's great, but we're far from having solved the protein folding problem. The question is, where does all this uncertainty in the predictions of proteins, of entire protein structures that then affect protein-protein interactions, play a role in your in
Well,
Rosetta?
let's see. I mean, I think, first of all, a lot of proteins contain disordered regions, which will not be predicted accurately. And then, protein structure is determined by, what's in the environment, like other proteins, small molecules, and so forth. it was like the question I was asked about predicting a oligomeric state. You know, that's not a very well posed problem if you're just given the sequence, because there might be multiple oligomeric states. So I would say the biggest uncertainty is maybe chemical composition. You know, what is present, you know, which chains are present, how many of them. That's certainly one large source of uncertainty. And then, of course, the networks are still not perfect. You know, they require, for native proteins, they require multiple sequences, usually at least 40 or so sequences from different organisms, because they're picking up on coevolution information. So I think there still is definitely room for improvement.
Okay.
Okay.
Yeah, sure.
I don't. And where do you see this improvement coming from?
I think the models will keep improving. I mean, we've been able to improve Rosetta Fold considerably over what we published earlier, and I think, you know, I think there will be the models currently don't really rely that much on sort of the detailed interactions between the side chains, so there are improvements there. I mean, it's like any method that they will get better over time.
Thank you.
Okay. Two general questions from me, if you still have time. The one is that I'd like to ask the landscape of next 10 years of the evolution of computer-based protein prediction and the effect of to the industry or social impacts. Could you give us some comments about that?
Right. Let's see. Well, so yes, so the first question is predicting the course of progress over the next 10 years. And the second is the the impacts on society. So as far as the first one goes, there's some people who are scientific visionaries who think they can see far ahead in the future. And my experience is that, particularly in rapidly moving fields like the one I'm in, if you can predict 3 months ahead, you're doing pretty well. So, you know, if I had heard the talk I gave today a year ago, I would have been extremely surprised. So, and it's fun to be in a field where you can't really predict what's going to happen, or because they're going to, you know, so I don't really want, I don't think I can predict the 10 years out. I think it's clear that we're in a time when you know, both structure modeling on the computer and protein design are increasing in power very, very rapidly. And I think it would be, I doubt if anyone could really make an accurate guess of where we'll be in 10 years. But I think I guess what I would, currently now in protein engineering, there have been, the primary approach has been random selection. So you make big libraries and you select for the molecules you want. And sort of doing things on the computer where you now do it based on principles hasn't been really as powerful. But I think what will happen is, I would guess 10 years from now, really the random library approaches will be sort of superseded by computational design approaches. But, you know, I think, you know, I can just only imagine, I mean, I think we'll be able to do really amazing things 10 years from now. But anyway, the field collectively. So as far as the impact there, I didn't talk about it today, but we've been designing proteins that are cancer therapeutics, anti-inflammatory therapeutics, and they have a lot of advantages. They're really small proteins. They're very stable. They can produce in large quantities. They can penetrate better into the body. And so those are currently in clinical trials. So in the next year, we're going to get readouts from clinical trials on designed therapeutics. Using that self-assembling nanostructure technology, my colleague Neil King has made coronavirus vaccines, which actually are pretty much at the end of phase 3 clinical trials and will be out in the world for use later this year. And he has a succession of new flu vaccines and RSV vaccines in the work. So what we're going to see in terms of impact is designed proteins in the clinic and as vaccines and therapeutics, you know, hopefully. You know, outside of the biomedical realm, I think there's a lot of really exciting potential for proteins as materials. For example, one thing I'm very excited about is biomineralization. So we have, you know, proteins that bone and tooth and seashell, which nucleate the deposition of inorganic materials. And if we could figure out how to design proteins to do that, then we'd have huge numbers of applications outside the material, outside the medical area. I talked a little bit about light harvesting. So I think we're going to first see the impact in the medical area, and then hopefully, a wide range of impacts outside of the medicine.
Great, thank you. The last question is very general, non-scientific. I would like to ask you to give us give young people, young researchers some advice or messages to do groundbreaking work like you did. So if you have any messages or advices, please give us comment.
Yeah, let's see. Well, that's a good question. I would say try and do what you're most excited about. I would caution people about, sometimes people worry about taking a big jump and they think it's better to be more conservative. But I think that if you try and take small steps, it also takes a lot of work, because everything takes a lot of work. And so you could be better off taking bigger steps. And so having a project, what you're working on, well, there's always risks in science. You just never know whether things are going to work. But I would say try to be more adventurous, take big steps. Also, you only live once, so you might as well try and kind of safe. There's not really any such thing as a safe project, because technology will change all the time. So I would say follow your passions and be adventurous, and be courageous. I think that would be my advice. And then try and find good mentors. I think that's really important. It's very hard to do science on your own. So collaboration is really important, and working with people that you like, who are sort of experts in the area that you're interested in. I think that's really, really important.
Yeah, thank you so much. So thank you again for excellent talk and taking your time in the early morning. And hoping to have you in person in the future sometime.
Yeah. Well, thank you very much for the invitation. Those were really excellent questions and discussion. So I enjoyed it very much. Thank you.